warm welcome to the Day KRANTI 2026, our 30-day National Agriculture Training Program. On the behalf of the Agri SAARTHI team, I warmly welcome to our respected speaker, Dr. Jagdish Prashad sir, Organizing Director Mr. Ajay Kasana sir, Chief Secretary of Agri SAARTHI, and all the enthusiastic participants present here today. My name is Pooja, and along with Ms. Simran, I will be guiding you through today's session. In the previous session, we discussed NEP 2020 and quality higher education in India, focusing on its current status, key initiative, and the way forward. Building on the importance of knowledge and practical implementation, today's session shifts our focus to the field level with an important agriculture topic. फसल का अवशेष कचरा नहीं बल्कि मिट्टी के लिए एक बहुमूल्य संसाधन है। इसी विचार के साथ मैं आप सभी प्रतिभागियों का आज के इस प्रशिक्षण कार्यक्रम में हार्दिक स्वागत करती हूं, जिसका विषय है क्रॉप रेजिड्यू मैनेजमेंट एंड वे फॉरवर्ड। क्रॉप रेजिड्यू मैनेजमेंट आज के समय में एग्रीकल्चर का एक बहुत ही इंपॉर्टेंट टॉपिक है, क्योंकि क्रॉप रेजिड्यू का सही प्रबंधन सॉइल हेल्थ सुधारता है, पॉल्यूशन को कम करता है और सस्टेनेबल फार्मिंग का बढ़ावा देता है। We are glad to have your active participation here, and we hope that today's training will provide you with practical and field level knowledge which you can easily apply in your farming practices. For the formal introduction of today's speaker, I now invite Ms. Simran. Ms. Simran, please.
Thank you, Ms. Pooja. It is my honor to introduce our esteemed resource person for today's session, Dr. Jagdish Prasad. Dr. Prashad sir is a noted agricultural research scientist currently working at Chaudhary Charan Singh Haryana Agricultural University, Hisar, in field of microbiology. He is an alumnus of CCSHAU Hisar itself. Dr. Prasad is currently working as assistant scientist and supervision of laboratory and field experiment related to microbial consortia and soil health. He has 22 years of teaching and research experience. He has an extensive experience in isolation, screening, and characterization of lingocellulitic fungi and bacteria. He has published several research papers and also is writer of a book in biodiversity and climate change trends in present context. Dr. Prashad also holds patent of microbial biodecomposer formulation for crop residue management and also contributed to Arjun SPP project also. He is the member of Microbiological Society of India and Indian Society of Soil Science. We are truly privileged to have Dr. Jagdish Prasad sir today with us. And with great respect, I invite sir to share his insights on crop residue management. Over to you, sir.
Hello. Hello.
Yes, sir, you are audible.
Hello.
Yes, sir, you are audible, sir.
Okay. So, welcome, respected dignitaries, honorable guest, esteemed member of this unit, and dear students. A very warm and pleasant welcome to all of you. It gives me immense pleasure to welcome you on the behalf of the organization committee on this program. I think it gave me immense pleasure to welcome you all on behalf of the organization committee to this program, and your presence here today has truly added grace and significance to the occasion. We are deeply honored to have such a distinguished person like Ajay and learned participants among us. This gathering has been organized with the objective to sharing the knowledge, exchangeable ideas, and encouraging meaningful discussion in our respective field. Such a platform not only enhance academic and professional growth, but also strengthen collaboration and mutual understanding. I would like to extend my sincere gratitude to our respected guests for sparing their valuable time to be with us today. It also thanks all participants for their enthusiastic presence and active involvement. And I am confident that today's program will be informative, inspiring, and fruitful to all, everyone present here. Once again, I warm welcome you all and wish this program every success. Thanks for giving time to me, and now I will start my PPTs. Can we start, sir?
Yes,
Please
sir.
share.
Please start.
Okay. So today we will discuss on role of microorganism in management of agriculture waste. Actually, you know that crop residue, there are so many crop residues, and you know that mostly people are not aware about the crop residue, what is crop residue and how we can use, and people have no knowledge about this, and some are ignorance about this crop residue, what are their physical or economic feasibilities are there, and time management. This one is also a important issue we will discuss. And you know that all the time you can see किसानों ने पराली न जलाने की शपथ ली, कटाई के बाद खाना प्रबंधन पर विशेष ध्यान देने की जरूरत, किसानों को खाना नहीं जलाने के प्रति जागरूक किया जाएगा। तो ये बहुत सारी आप कटिंग्स जो हैं वो देखते होंगे हर रोज पेपर में, कहीं ना कहीं आपके यहाँ आती रहती होंगी। तो आज इसके बारे में हम पूरा विस्तार से जो है वो डिस्कस करेंगे कि कैसे हम इनको जो है, इस प्रॉब्लम को, जो ये बहुत बड़ी प्रॉब्लम है, कैसे इसको मैनेज करें। देयर आर सो मेनी प्लान्स आर देयर, एंड इन विच वन of them is central plant to boost 2G ethanol production, set up enzyme manufacturing facilities to reduce imports. So there are many problems are there behind this crop residue management, and many companies are engaging in this program. Particularly, you know that agriculture waste mainly consists of organic waste, inorganic waste, hazardous waste, post-harvest agriculture waste, agro-industrial residues, and among the organic waste, basically plant residue, animal waste, food waste, and there are so many chemical waste, plastic waste, metallic waste, pesticide and herbicide, veterinary, pharmaceutical, and heavy metals are there. So all these are basically included in agriculture waste. Actually, crop residue is the left out material after cultivation of crop that has been harvested, and you can see these are basic crop residues are there, and among them rice residue is the main problem. You know that there are field residue, and field residue are non-economic materials left in the agriculture field or orchard after crop has been harvested. These residue includes stalks, stubbles, leaves, seed, pods. The residue can be plowed directly into the ground, or it can be used as a mulch on the field. Process residue or material left after crop is processed for unusable resources. This includes husk, seed, bagasse, molasses, roots. It can be used as animal fodder or soil amendment. सो आप देख रहे हैं कि यहाँ पर एग्रीकल्चर रेजिड्यू की हम बात करते हैं, तो फील्ड जो हमारा रेजिड्यूज होते हैं वो बेसिकली जो लेफ्ट ओवर जितने भी हमारी फसल की कटाई के बाद जो भी बचते हैं उनको हम एग्रीकल्चर रेजिड्यू में मानते हैं। और दिक्कत कहाँ आती है कि लोग ये समझते हैं कि अवेयरनेस नहीं है, इग्नोरेंस है, लोग अवेलेबल नहीं है, नॉलेज नहीं है, और इन्हीं कारणों से जो रेजिड्यू मैनेजमेंट हमारे लिए एक क्रॉप रेजिड्यू मैनेजमेंट एक प्रॉब्लम है। और सबसे बड़ी प्रॉब्लम जो है वो पैडी फील्ड के अंदर फार्मर यूजुअली क्या है उसको जला देते हैं। बर्निंग इश्यू जो है मेजर इश्यू है। और अगर हम रेजिड्यू की बात करें तो रेजिड्यू के अंदर मोस्टली जो है जनरेशन इंडिया के अंदर, अगर हम वर्ल्ड के अंदर बात करें, कहीं भी अगर सोचते हैं तो हाईएस्ट जो रेजिड्यू जनरेशन है, आप देख रहे हैं यूपी, पंजाब, हरियाणा, महाराष्ट्र, यहाँ पर सबसे ज्यादा क्रॉप रेजिड्यू की प्रॉब्लम आती है। और इनमें से मोस्टली जो क्रॉप्स आती हैं वो सीरियल जनरेटेड हैं, और उसके बाद फाइबर, ऑयल सीड, पल्सेस, शुगर केन, ये सबसे बड़ी प्रॉब्लम है। शुगर केन की भी बहुत बड़ी प्रॉब्लम है। अगर हम बात करें सीरियल क्रॉप की, जिसमें राइस, व्हीट, मेज, मिलेट्स, दे कंट्रीब्यूट मोर देन 70%, एंड अमंग देम राइस कंट्रीब्यूट 34%, wheat rank second in the world, 22% of the crop residue, and more than 683 million tons of crop residue of different crop are produced, of which major part is used as a fodder, fuel, and in various industrial sector. But despite this, about 170 metric ton of surplus crop residue are available around the country, as reported by TIFAC 2018, Technology Information Forecasting and Assessment Council. Crop residue, actually they are consist of lignocellulosic material, and we can say that they are lignocellulosic in nature. You know lignocellulose. Lignocellulose mean that contain high portion of cellulose, hemicellulose, and lignin. Basically, these are the three components: cellulose, hemicellulose, and lignin. और हम बात करेंगे लिग्निन, क्योंकि इसमें लिग्निन है तो इसलिए हम इसको कहते हैं लिग्नोसेलुलोजिक मटेरियल। और सारे के सारे जो लिग्नोसेलुलोलिजिक लिग्नोलिटिक जो मटेरियल हैं, सारे के सारे लिग्नोसेलुलोज से बने हुए हैं। और सबसे ज्यादा इसमें क्या होती है, हेमी ये सेलुलोज, हेमी सेलुलोज और लिग्निन। अगर हम लिग्निन की बात करें तो लिग्निन बड़ी, अगर हम बात करें डिग्रेडेशन की, तो लिग्निन डिग्रेडेशन में बहुत टाइम लगता है। अगर राइस की बात करें तो राइस में आपके लिग्निन, हेमी सेलुलोज और सेलुलोज सबसे ज्यादा होती है। इसी कारण जो है ना, ये जल्दी से डिग्रेड नहीं होता। हम कहेंगे कि ये जल्दी से जो है, मोस्टली अगर हम बात करके चलें कि डिग्रेडेशन कैपेसिटी की माने, तो लिग्निन को कम से कम 1 साल लग जाता है इसको कंप्लीटली अगर हम उसको डिग्रेड करें। ये जल्दी से डिग्रेड नहीं होता। और सबसे बड़ी हमारी प्रॉब्लम जो है वो सेलुलोज, हेमीसेलुलोज और लिग्निन की ही है। और जितने भी लिग्नोसेलुलोजिक जो हमने एग्रीकल्चर रेजिड्यू की बात की, अ, ये ही जो है हमारे लिए सबसे बड़ी प्रॉब्लम क्रिएट करते हैं। ये जल्दी से क्यों नहीं डिग्रेड होते? अगर हम इसकी कंस्ट्रक्शन की बात करेंगे, हम कि कैसे इनकी जो स्ट्रक्चर है वो कैसे होती है, वो स्ट्रक्चर मैं आपको आगे बताऊंगा। लेकिन हम बात करें इसके अलावा अभी जो दूसरे मटेरियल्स, आपने देखा कि कई बार हम खेतों में जाते हैं या कहीं भी एग्रीकल्चर रेजिड्यू की बात करते हैं, एयर पोल्यूशन की बात करते हैं, क्रॉप रेजिड्यू मैनेजमेंट की बात करते हैं। तो एयर पोल्यूशन की अगर हम बात करें, या हमारे जितने भी कैटल्स हैं, कैटल या कैटल भी हमारे लिए एक बहुत बड़ी समस्या है। कैटल डंग, अगर हम कहूँ कहीं बात करें गौशालाओं की, जो हमारे यहाँ बहुत सारे कैटल्स हैं या पशु हैं, उनके जो गोबर की बात करें हम, जो कैटल डंग है, तो उसमें काफी ग्रीन एमिजन होता है। ग्रीन एमिजन मोस्टली जो ग्लोबल वार्मिंग की जो गैसेज होती हैं वो अवेलेबल होती हैं, चाहे वो ग्रीन हाउस गैसेज जितने भी है, कार्बन डाइऑक्साइड हो या दूसरी गैसें हैं, नाइट्रस ऑक्साइड है, कार्बन मोनोऑक्साइड है, मीथेन है, ये सबसे बड़ी हमारे लिए एक प्रॉब्लम है। और जहां तक हम बात करें, हम हमारी सोयल की बात करें, अगर सोयल की बात करते हैं तो इंडियन सोयल अगर हम बात करें, उसके अंदर जो मैक्रो एंड माइक्रो न्यूट्रिएंट्स की बात करते हैं हम लोग, तो उसमें 17 तरह के एलिमेंट जो हैं प्लांट्स को चाहिए। और वो 17 तरह के एलिमेंट्स जो हैं मैक्रो एंड माइक्रो न्यूट्रिएंट्स होते हैं। और ये मैक्रो एंड माइक्रो न्यूट्रिएंट्स कहाँ से आते हैं? क्योंकि नेचर का एक अपना सिस्टम होता है कि हमारे जो रेजिड्यूज हैं उसमें मैंने बताया कि रेजिड्यूज में सेलुलोज भी होता है, हेमीसेलुलोज भी होता है, लिग्निन भी होता है। इसके अलावा जो है स्मॉलर अमाउंट में जो है पैक्टीन, प्रोटीन और दूसरे एक्सट्रैक्टिव्स जो होते हैं, वेदर दे आर सोल्यूबल और नॉन सोल्यूबल, नॉन स्ट्रक्चरल सुगर्स, नाइट्रस मटेरियल, क्लोरोफिल, वैक्सीज एटसेट्रा। इवन देन कई बार सोचते हैं कि हमने आग लगा दी तो आग लगाने के बाद में भी जो कार्बन आएगी, ये तो बहुत सारे मिथ और मिसकंसेप्शंस हैं हमारे सामने। पर एक हमारे लिए प्रॉब्लम है, मैंने सबसे पहले स्लाइड में कहा कि इग्नोरेंस नॉलेज का, ये बहुत सारे ऐसे इश्यूज हैं जो हमें सॉल्व करने हैं। क्योंकि दिक्कत कहाँ आती है, जनरली अगर हम बात करें सेलुलोज, हेमी सेलुलोज और लिग्निन की बात करें, तो लिग्निन अगर हम बात करें तो लिग्निन तो है 13 से 14% जो है आपके लगभग राइस रेजिड्यू में मिलती है। इसके अलावा हेमी सेलुलोज और सेलुलोज जो है, दे कैन कॉन्स्टिट्यूएंट्स अप टू 70 to 80%। of agriculture waste, but in some cases up to 99% of cellulose, 85% of hemicellulose, and 40% of lignin have been found in agriculture residue. That is the main problem. And how we can solve this problem? That is the issue. Whether they can degrade themselves, or we have to enrich some microbes that can easily degrade them in the soil. And there are so many cycles are there. आपने देखा होगा कि साइकिल्स होते हैं बहुत सारे, चाहे आपने पढ़ा भी होगा, कार्बन साइकिल, नाइट्रोजन साइकिल, वाटर साइकिल। ये साइकिल क्या है? ये सारे नेचर की देन है। अगर हम बात करें नाइट्रोजन की, बात है 78% नाइट्रोजन इज प्रेजेंट इन एटमॉस्फेयर, बट प्रॉब्लम इज दैट plants are unable to directly consume that nitrogen in elemental form, so it has to first rate, and it can be done by with the help of microbes. So microbes can play a crucial role in our agriculture system. Now the problem is that we have already discussed that our soil has carbon content which is very small. Small mean, अगर हम हरियाणा की बात करें तो 0.5%, 0.5 से भी कम आपको कार्बन मिलेगा। और ये कार्बन जो है वो कहाँ जाती है? एटमॉस्फेयर में कैसे उसको हम ट्रैप करें, कार्बन को सिक्वेस्ट्रेशन कैसे करें, वो भी हमारे लिए एक समस्या है। कि आपने मान लीजिए, हेलो
डॉक्टर साहब, एक्सक्यूज मी सर, सॉरी टू इंटरप्ट, बीच बीच में आपकी आवाज ना बहुत लो हो रही है, बच्चे बोल रहे हैं कि सुनने में उन्हें दिक्कत आ रही है। इंटरनेट तो आई थिंक सही आ रहा है, तो हाँ बीच बीच में पता नहीं क्यों
इंटरनेट तो
बहुत।
सही है सर।
हाँ बिल्कुल सही है
चलो
सर।
कोई नहीं, मैं ट्राई करता हूँ कुछ और।
हाँ जी प्लीज।
सबसे बड़ी दिक्कत कहाँ आती है? आवाज आ रही है सर मेरी? हेलो।
यस सर, यू आर परफेक्टली ऑडिबल सर।
तो सबसे बड़ी प्रॉब्लम कहाँ आती है कि हमारा जो सोयल का कार्बन जो है वो बहुत कम है। तो सबसे बड़ी आज की हमारी प्रॉब्लम जो है वो कार्बन की है, कि कार्बन को हम कैसे जो है रोकें, sequestration कैसे करें। बहुत सारे वैज्ञानिक लगे हुए हैं, चारकोल पेस्ट हैं कि कैसे उसको चारकोल डाल करके उसको रोक सकें, ताकि वो जो है पौर होते हैं उसमें काफी वो कार्बन को सिक्वेस्ट कर लेता है। अब दिक्कत क्या आती है कि हमने यूज किया, हम कंपोस्टिंग करते हैं, हम और कुछ भी करते हैं, तो बहुत सारी हमारे सामने ये समस्याएं रहती हैं। देखिए, अगर आप बात करें सेलुलोज, हेमी सेलुलोज और लिग्निन की स्ट्रक्चर की बात करें, यू कैन सी फ्रॉम दिस स्लाइड दैट actually cellulose, hemicellulose, and these lignin, they are mainly they are polymer of glucose, aromatic polymers and sugars. And you can here see that cellulose, hemicellulose, lignin structure, and you will find that these are the polymer of glucose. Polymer of glucose in the sense की सारी ग्लूकोज के पॉलीमर हैं। अगर हम ग्लूकोज की बात करें सेलुलोज में, ग्लूकोज की जो स्ट्रक्चर हमने दिखाई है नीचे आप देख रहे हैं, इसमें एक लिंकेज होती है, बीटा 1,4 लिंकेज होती है, और बीटा 4 लिंकेज की एक बहुत लंबी चेन होती है, और वो आपस में मिलकर के सेलुलोज बनाते हैं। इसी तरह से अगर हम बात करें हेमी सेलुलोज की, तो उसमें क्या है, C5 और C6, यानी यहाँ पर ग्लूकोज, जाइलेनोज, रेबिनोज, मैनोज, ग्लैक्टोज, ये मिलकर के एक लंबी चेन बनाते हैं। उसी तरह से बॉन्डिंग होती है। अगर लिग्निन की बात करें तो इसमें बेसिकली क्या होता है, कोनिफेराइल अल्कोहल, पैरा क्यूमेराइल अल्कोहल एंड सिनापाइल अल्कोहल, और इनको तोड़ना बड़ा कठिन होता है। इवन देन माइक्रोब्स भी जो हैं जल्दी से नहीं तोड़ पाते। कई बार डिस्कस होता है कि पूसा ने एक डिकम्पोजर किया है, पीछे में कर देता है, में कर देता है, क्या हम इसको मान के चलते हैं। लेकिन अगर हम लिग्निन की डिग्रेडेशन की बात करें तो डिग्रेडेशन बड़ा स्लो प्रोसेस है, और ये होता है माइक्रोब्स की सहायता से। माइक्रोब्स, लिग्नोसेलुलिटिक माइक्रोब्स की हम बात कर रहे थे। लिग्नोसेलुलिटिक का मतलब है कि लिग्निन, सेलुलोज, हेमीसेलुलोज, उसको हम कैसे तोड़ें और उसको तोड़ने के लिए कौन जिम्मेवार है। अगर हम बात करें सबसे ज्यादा, तो जो माइक्रोब्स जो क्रूशियल रोल अदा करते हैं वो एंजाइम सिक्रेट करते हैं। एंजाइम मीन, जब आप पॉलीमेरिक चेन होती है, एक लंबी चेन होती है, मान लीजिए एक रस्सी है बहुत लंबी है, उसको हम टुकड़ों में कैसे तोड़ेंगे। जैसे एंजाइम जो होते हैं वो एक कैंची की तरह काम करते हैं, जस्ट लाइक सीजर। जैसे कैंची काटती है किसी रस्सी को, उसी तरह से ये एंजाइम जो है माइक्रोब्स सिंथेसाइज करते हैं। और ये माइक्रोब्स यूजुअली जो एंजाइम सिक्रेट करते हैं वो सारे के सारे एंजाइम जो है हाइड्रोलिटिक नेचर के होते हैं। और वो एंजाइम आपके कॉम्प्लेक्स मटेरियल को, जो पॉलीमर चेन होती है, लंबी चेन होती है, उसको छोटे छोटे टुकड़ों में तोड़ देते हैं। फिर दूसरा एंजाइम होता है, फिर उसको तोड़ता है, तीसरा फिर तोड़ेगा, चौथा फिर तोड़ेगा, और लास्ट में क्या है, कॉम्प्लेक्स मटेरियल जो है वो सिंपल के अंदर चेंज हो जाएगा। और इस प्रोसेस में आपके खास तौर पे जो एंजाइम इन्वॉल्व होते हैं, सेलुलोज को तोड़ने के लिए हमारे पास सेलुलेज एंजाइम होता है, जिसको सीएमसीएज कहते हैं हम, हेमी सेलुलोज। इसको हम जो है फिल्टर पेपरेज एंजाइम कहते हैं, उसकी सहायता से ये टूटता है। और लिग्निन की बात करें तो हम लिग्निन पेरोक्सीडेज एंजाइम्स होते हैं, लैकेज एंजाइम्स होते हैं। और ये एंजाइम सारे के सारे माइक्रोब्स, और माइक्रोब्स की हम बात करें तो माइक्रोब्स में बैक्टीरिया और फंजाई दोनों आएंगे आपकी। अब बात करें तो बैक्टीरिया की, बैक्टीरिया भी जल्दी से तोड़ देते हैं क्योंकि वो भी लिग्नोसेलुलिटिक होते हैं, यानी वो एंजाइम सिक्रेट करते हैं जो इन कॉम्प्लेक्स को तोड़ने में हेल्प करते हैं। बट प्रॉब्लम कहाँ आती है कि माइक्रोब्स जो है वो वेदर जिस नेचर में आपने उसको दिया, जहां पे आपने उसको इनरिच किया, मान लीजिए एक सॉइल है, उस सॉइल में हम अगर हम बॉर्डर के ऊपर, पीपल तो सारे हमारे लोग तो एक जैसे हैं, लेकिन बॉर्डर पे हमारी सेना होती है, फौजी होते हैं ना, इसी तरह से हम उन माइक्रोब्स को आइसोलेट करते हैं। आइसोलेट करके उनकी प्रॉपर्टीज जो है वो चेक करते हैं कि वो कौन कौन से, उन एंजाइम का पूरा स्क्रीनिंग करते हैं कि वेदर these microbes they can synthesize these enzymes or not. बट अगर हम फंजाई की बात करें, फंजाई जो होती है वो एक्चुअली फिलामेंटस नेचर की होती है, बहुत जल्दी प्रोलिफेरेट होती है, फास्टिडियस होती है नेचर के अंदर। फास्टिडियस मीन वो उनकी ग्रोथ बहुत जल्दी होती है, और वो मैक्सिमम जो एंजाइम जो है वो ज्यादा मात्रा में स्प्लिट करती है। और बहुत सारी फंजाई हैं जिनके नाम है, कॉमनली आप सुनते भी रहते होंगे, एस्परजिलस भी नाम सुना होगा आपने। एस्परजिलस मेनली क्या है, कई बार आप घर में प्याज देखते हो, प्याज के ऊपर ब्लैकिश ब्लैकिश स्पोर्स आ जाते हैं। तो स्पोर क्या है, स्पोर एक्चुअल में क्या है, वो सारे के सारे स्पोर जो हैं एस्परजिलस के होते हैं। अब एस्परजिलस में भी स्पीसीज बहुत सारी होती हैं, एस्परजिलस फ्लेवस है, एस्परजिलस नाइजर है, एस्परजिलस फ्यूमिकेटस है, ओराइजी है, और है। इसके अलावा कुछ ऐसी और फंजाई जिसके आपने नाम सुना होगा, ट्राइकोडर्मा आपने सबने नाम सुना होगा, ट्राइकोडर्मा, हर्जेनियम। ये जो है, इसके अंदर मोर देन 95 पेप्टाइड्स होते हैं, शॉर्ट पेप्टाइड्स होते हैं, सेकेंडरी मेटाबोलाइट्स होते हैं, वो सिंथेसाइज करती है। इसके अलावा सारे के सारे एंजाइम जो लिग्नोसेलुलिटिक होते हैं, लिग्नोसेलुलिटिक का मतलब होता है वो एंजाइम जो लिग्निन, सेलुलोज और हेमीसेलुलोज को तोड़ दे। वो सारे के सारे एंजाइम जब मैंने बताए, सेलुलेज, हेमीसेलुलेज और लिग्निन पेरोक्सीडेज, लैकेज, ये एंजाइम है। अगर ये एंजाइम, ये फंजाई जो है तो सिक्रीट करती है, और वो एंजाइम जो है अब क्या करेंगे, उनके लिए हमारा जो रेजिड्यू है वो दैट एक्ट एक्ट एज ए सबस्ट्रेट, कि वो उसके लिए खाने का काम करेंगे, कि वो उस फंजाई उस पार्टिकुलर रेजिड्यू को खाएगी, रेजिड्यू को वो पार्टिकुलर को खाएगी तो उसका अपना जीवन होगा। अब वो क्या करेगी, उसका डिकम्पोजिशन करेगी, डिग्रेडेशन करेगी। अब डिग्रेडेशन दो तरह से, एक अनएरोबिक है, एक एरोबिक भी होता है। आपने देखा होगा कंपोस्टिंग होती है, जब हम गोबर की बात कर रहे थे तो गोबर के अंदर तो बहुत सारे हैं, और वो हेलोजेनिक बैक्टीरिया एरोबिक होते हैं। आपने देखा होगा बायोगैस में क्या होता है, बायोगैस में यूजुअली तो है गोबर, और उसके साथ साथ आपका जो डोमेस्टिक रेजिड्यू है वो हम डाल देते हैं। और वो अनएरोबिक कंडीशन के अंदर, उस चैम्बर के अंदर जो है, उस जो लिग्नोसेलुलिटिक मटेरियल्स होते हैं उनको क्या करेंगे, वो उनके लिए खड़ा खाएंगे, बदले में वो गैस रिलीज करेंगे, बदले में आपकी मिनरलाइजेशन शुरू करेंगे। धीरे धीरे जो आपके ऊपर में एग्रीकल्चर रेसिड्यूज है उसको तोड़ेंगे। तोड़ेंगे कैसे, वो धीरे धीरे तोड़ते रहेंगे कॉम्प्लेक्स मटेरियल को, शॉर्ट में, फिर उससे छोटा, फिर उससे छोटा, और अल्टीमेटली जो मिनरलाइजेशन होगी उसमें क्या होगा कि आपके जो मिनरल्स हैं, जो मैंने कहा कि 16 तरह तरह के एलिमेंट हैं जो प्लांट की ग्रोथ के लिए चाहिए, वो इजिली जो है उस आपकी जो मैन्योर होगी, सलरी होगी, उसके अंदर आ जाएंगे। चाहे वो कंपोस्टिंग, हम सलरी की बात करें, गोबर गैस की बात करें, या हम कोई भी डिकम्पोजर नाम सुना होगा कि डिकम्पोजर क्या होता है। बहुत चीजें आप कहीं पशु आपने देखे होंगे आप जहाँ तो मरने के बाद में 15 20 दिन 25 दिन के बाद उसका क्या होता है, पहले एक फॉल स्मेल आती है, यूट्रिफिकेशन होता है, उसकी प्रोटीन है और है, वो सब धीरे धीरे टूटते रहते हैं। और टूटने के बाद में लास्ट मिनरल्स है उस सॉइल के अंदर इनऑर्गेनिक फॉर्म में आ जाते हैं। फिर वो कहीं से धूल हो मिट्टी हो कहीं से उड़ करके या जैसे भी है आपके खेत में या आपकी सॉइल के अंदर वाटर के द्वारा किसी भी मीडियम से चले जाते हैं और प्लांट फिर उसको दोबारा से अब्जॉर्ब कर लेते हैं। और अब्जॉर्प्शन के बाद दैट बिकम ए पार्ट ऑफ दैट प्लांट एंड इन दिस वे द प्लांट जो है दे गेट न्यूट्रिशन और इंक्रीज करेंगे, ग्रोथ करेंगे। तो इस तरह से एग्रीकल्चर, हम रेजिड्यू की बात करें तो एग्रीकल्चर रेजिड्यू एक नहीं है, खेत के अंदर जितने भी अवशेष हैं जो हमारे बचते हैं उसको हम बड़ा वो लेते हैं, एक हल्के में ले लेते हैं कि भाई ये तो कुछ नहीं है, कुछ काम का नहीं होता। एक्चुअल में ऐसी बात नहीं होती। ये सबसे बहुमूल्य चीज है और इसको यूटिलाइज किया जा सकता है। बहुत सारे हमारे पास वेज हैं, उस तरीके से हम इसको, अगर आप बात करें कि हम अभी जो डिस्कस कर रहे थे कि रेजिड्यूज के अंदर जो है कितने प्रकार के किस किस तरह के कंटेंट जो है वो अवेलेबल होते हैं, तो ये हमारे पास एक लिस्ट है कि जितने भी लिग्नोसेलुलिटिक मटेरियल हैं, जैसे बगासे है, पीनट है, राइस हल है, राइस हस्क, राइस स्ट्रा, सुगर केन लीफ, स्टॉक, सोरगम, हार्टवुड स्टेम, सॉफ्टवुड, नट सेल्स, कॉर्न, paper, wheat straw, sorted refuse, leaves, cotton seed, hair, newspaper, waste paper from chemical pulp, primary waste, solid cattle manure, coastal, ये जो है बर्मुडा ग्रास, स्विच ग्रास, वाइन वेस्ट। अब इनमें सब में इस लिस्ट में आप देखें कि हर जगह डिटेल्स है, हेमिसेलुलोज, लिग्निन। तो हर एक में देखिए आप कंटेंट जो है वेरिएबल है। अब बगासे में 20% तक होती है, पीनट के अंदर 35 तक होती है। अगर हम बात करें नट सेल्स की, 40% तक जो है लिग्निन होती है। और लिग्निन जो है सबसे मुश्किल इसको तोड़ना है, इसके डिग्रेडेशन में बहुत टाइम लगता है, ये बड़ा कॉम्प्लेक्स है। आपने पिछली जो है स्लाइड में देखी, इसकी स्ट्रक्चर देखिए आप, इसमें 3 चीजें होती है, कोनिफेराइल और ये पैरा क्यूमेराइल अल्कोहल, सिनापाइल अल्कोहल। ये कॉम्प्लेक्स फैक्टर है और इन स्ट्रक्चर को तोड़ने में थोड़ा सा वक्त लगता है, टूट जाती है, क्योंकि प्रकृति का एक नियम है कि प्रकृति ने जिस चीज को पैदा किया प्रकृति उसको विनाश भी करती है। तो विनाश का मतलब है, चाहे वो नाइट्रोजन का चक्कर हो, कार्बन का हो, चाहे न्यूट्रिएंट साइकिल्स हो, ये साइकिल एनवायरनमेंट में चलते रहते हैं। और एक सेल्फ सस्टेनेबल जिसको हम कहते हैं मैकेनिज्म जो है वो चलता है। और ऐसे मैकेनिज्म जो है उसी की वजह से सर्वाइवल होता है हमारा। अब देखिए आप लिग्नोसेलुलोजिक का biomass, यहाँ पर हम राइस की बात कर रहे हैं, ऊपर वाली picture देखिए आप, लिग्नोसेलुलोज बायोमास है। और खासतौर पे सेलुलोजिक का जो बायोमास राइस के अंदर कैसे उनकी स्ट्रक्चर होती है, देखिए आपको दिखाई दे रहा है ना, ये सेलुलोज जो पाइप स्ट्रक्चर मैंने खड़ी की हुई है, ये सेलुलोज है। और फिर उसको एक लकड़ी है जैसे मान लीजिए ये, और इसको किसी बंडल से हमने बांध रखा है, वो आपका हेमिसेलुलोज है। और फिर उसको और एक हाई तरीके से जो है पूरा का पूरा रैप किया हुआ है, वो क्या है आपकी लिग्निन। तो सबसे पहले हमें किसको तोड़ना है, लिग्निन को। उसके बाद हेमिसेलुलोज सेलुलोज टूटेंगे और धीरे धीरे इससे ग्लूकोज अल्टीमेटली मीन ये ग्लूकोज तक चले जाएंगे। और ग्लूकोज के बाद फिर मिनरलाइजेशन होगा। तो ये एक बेसिक स्ट्रक्चर होती है। आई थिंक ऑल ऑफ यू आर अवेयर विद दिस स्ट्रक्चर एंड यू कैन सी how these lignocellulosic biomass, you can see here that this one is lignocellulosic biomass and basically cellulose which is mainly consist of ग्लूकोज सब यूनिट। ये देखिए एक ग्लूकोज, दो ग्लूकोज, तीन, चार, पांच है ना ये, और इस तरह से एक लंबी चेन जाती है। और इनके बीच में जो बॉन्ड होता है ना आपस में, ये देखिए एक ग्लूकोज, दो ग्लूकोज, और ये एक लाइन दिखा रहे हैं हम, ये क्या है, ये आपके बीटा 1,4 लिंकेज होती है। इसी लिंकेज को जब एंजाइम तोड़ेंगे तभी इसके टुकड़े होंगे। दूसरे में देखिए आप हेमिसेलुलोज के अंदर, अब ये ब्रांच स्ट्रक्चर है। और तीसरी अगर हम बात करें लिग्निन की, तो लिग्निन के अंदर जो बॉन्ड होते हैं, मैंने तीन चीजें बताई थी आपको, पी क्यूमेराइल, सिनापाइल और कॉनिफेराइल। ये स्ट्रक्चर जो है बड़ी हार्ड होती हैं और इनको तोड़ना बड़ा मुश्किल होता है, तो ये थोड़ा सा टूटने में टाइम लेती है। इसीलिए लिग्नोसेलुलिटिक जो एंजाइम इस पे काम करते हैं वो थोड़ा सा स्लो करते हैं। लेकिन एक बार अगर ये स्ट्रक्चर टूट जाए, आप देखिए अगर सबसे बाहर की स्ट्रक्चर अगर हमारी टूट जाती है, ये वाली स्ट्रक्चर टूट जाए, तो इनको फिर तोड़ना बड़ा इजी हो जाता है। और इसके बाद दूसरे कंपोनेंट जो है दे कैन इजिली ब्रोकन डाउन। सर आवाज आ रही है ना? हेलो,
यस सर, इट इज
आ
ऑडिबल
रही है?
डॉक्टर।
समझ में आ रहा है?
हाँ जी प्लीज।
और सबको सबको समझ में आ रहा है ना?
हाँ जी सबको समझ में आ रहा है, बाकी थोड़ा सा अगर आप चाहें तो इंग्लिश में कंटिन्यू कर सकते हैं वो।
तो,
हाँ
अच्छा
जी
ठीक है,
प्लीज।
ठीक है सर।
हाँ जी डॉक्टर
So mainly crop residue is a good source of nutrients and all essential components which are available for our which are required for our agriculture system ecosystem. They are present in this residue and they can be released from these biomass slowly and slowly, just like such as nitrogen, phosphorus, potash, etc. And you can say that these are the macro and micronutrients which are required for the growth of the plants. Basically, these are the macronutrients which are required in large amounts, such as nitrogen, phosphorus, potassium, sulfur, magnesium, and calcium. And these are micronutrients which are required in very trace amounts. And if they are available in our soil or in our manure, they can easily be absorbed by the root of the plant, and even then they help in the soil health, maintenance of soil health, and they can make soil as a good texture in nature, so that plant can take these nutrients from root and others such as water, carbon dioxide, and light. They can take from our atmosphere. And in this way, these agriculture residue help in the maintenance of soil health. So there are so different components are present in our residue, and they can be utilized as a nutrients for the plant growth, and they are very helpful in the soil health maintenance. So, but the main problem that is in our state, particularly in Haryana and adjoining state Punjab and UP, they are causing the main problem and the burning issue, that is the burning of these residue, particularly rice residue. When we burn these residue, there are so many and such as the loss of carbon, emission of greenhouse gases, deterioration of the soil quality, loss of soil flora and fauna, loss of biodiversity, and so many health hazards are there, such as pneumonia and other disease. The people suffering from these different diseases, uh, they are unable to survive in such a condition when a when air quality index goes up to the 300 and more. So these are the main problem behind burning. And burning actually, when we burn any agricultural residue, particularly rice residue, they can emit more amount of carbon dioxide, more than 70% carbon monoxide, methane, and nitrous oxide. All these are green. We can say that they are the global warming. They produce global warming, and these gases particularly affect human health. They can degrade air quality. It can causes accidents and reduce the visibility. Even then, atmospheric pollution they will create, and such a huge burning causes many losses to human health. Apart from the nutrient loss, some properties of the soils they are also degraded, and even then they can increase the soil temperature, pH, moisture, phosphorus, and organic matter are greatly affected due to the burning. So burning, even then, main problem that burning can cause, that is reduction in the soil microbial organism. or microbes, and we can when again and again repeat the burning process. So after a time will come and microbial population, beneficial microbial organisms, they are reduced in our soil, and it can cause or reduce soil carbon and nitrogen, and potentially mineralizable nitrogen in the upper soil layer. There are so many agencies like NASA also have highlighted the problem of burning in the North Indian states. You can see here that some fire detection captured by the infrared imagery that is from NASA. These are the area where burning is the main problem. Now the question is how we can manage crop, that is crop residue management. You know, there are so many crop residues are there, and they can be processed. There are some processed residues such as oil cake, seed residue, husk, bagasse, molasses, root waste, etc. So they can be managed either by microbial process, and microbes are mainly used for the production of enzymes, that is lignocellulosic enzyme, ethanols, organic acid, antioxidants, antibiotics, etc. That can be done with the help of the fermentation. And mainly microbes which are involved in this process are yeast, bacteria, fungi, actinomycetes, etc. So they can change these residue into usable products, and sometime compost, mushroom beds, biofertilizer, green manures, etc. So in this way, we can manage these crop. So we can see here that microbial waste management is one of the critical management system in which bacteria alone, fungi alone, or sometime when bacteria and fungi, they are mixed cultured, they make some consortium, and these consortium help in the management of these agriculture residue. And you can see here that composting is one of the process in which we can compost these agriculture residue into a dark and gray colored manure, which can contain high amount of humic and fulvic acid, and these humic and fulvic acid mainly they help in the maintenance of the soil structure. And you can see that composting is a complex process. I cannot explain all these things, but you can here I cannot explain, because composting is one of the major process which can take some time to complete this process. And there are four main phases are there. Phase one, mesophilic phase, in which some mesophilic microbes are there, which can grow up to 40 degrees centigrade temperature, or we can say that we have microbes which can survive in 20 to 40 degrees centigrade temperature, where they perform their activities. And after that, second phase is thermophilic phase, and then mesophilic, and last one is the mesophilic phase is there, that is the maturation phase. And phase second, that is thermophilic phase, in which temperatures rises up to 80 to 100 degrees centigrade. And in this phase, what happened, the harmful microbes, pathogenic microbes, uh, they can be killed with the help of the high temperature. Then again, mesophilic microbes, they grow again and become active, and ultimately it enter into the next phase, that is the maturation phase, in which there is a formation of humic and fulvic acid, and that humic component is the major component which can increase the water holding capacity and improve the structure of soil along with soil components. So composting is a major process where microbes can play an important role. Decomposition process is of two types, either aerobic or anaerobic process are there. Anaerobic decomposition, if you see in first diagram, agriculture waste, food waste, and biosolids or organic waste are there. We can have some digester, anaerobic digestion, digestion system is there, where anaerobic conditions are maintained, and in these anaerobic digester, biogas is produced. You can our biogas plant, where we can maintain anaerobic conditions. In the similar way, this anaerobic digester actually help in the formation of biogas, which can be used for electricity generation, thermal, or as fuel. But here, solid part, after harvesting or processing of solid or liquid the effluents, either work as a compost or animal bedding, we can use them, or liquid product are there, organic fertilizer are there. So anaerobic digestion of lignosulfuric biomass, which can produce electricity, thermal fuel, etc. These are the basic steps. You can see that anaerobic digestion of lignocellulosic biomass, either from marine, forecast, agriculture, etc. And then, first of all, we identify these feedstock, then go for pre-treatment, either acidic or alkaline treatment is there. After pre-treatment, what happens? Hydrolysis will occur either with the help of acidogenesis or acetogenesis. Then ultimately methanogenic microbes come into play their role, and these methanogenesis process produce biogas, and cleaning help in the cleaning, upgrading, either can be used as a fuel or chemicals in the similar way, digestive post treatment that act as a fertilizer. So these are the main process you can see, or these process, what is hydrolysis, acidogenesis, and acetogenesis, and methanogenesis. These are the main step of anaerobic digestion. Now, another question is that how these solid state fermentation, that is the next step. Biofertilizer and bioremediation, waste to energy conversion. How we can convert these waste into valuable product, such as biofuels, bioplastic, biochemicals, and animal feeds from these waste. You can see this structure, how these some microbes they can be helpful in the formation of biofuel, such as biodiesel, bioethanols, and they have the capacity to convert these agriculture waste, particularly lignocellulosic biomass, into bioethanol or biodiesel. Then bioplastic, biochemicals, and animal feeds that can also be produced from these microorganisms, either by solid state fermentations. This one is the international. Now the question is that you know that rice can be managed either by soil incorporation, mulching, or we can remove these residues from our field, or either by burning. But burning has so many problems we have already discussed. What we have done in the last 5 or 6 years, we make a consortium of different organisms, and these consortia that can be multiplied, or we have a liquid based microbial product, even then compost or microbial inoculants, they can also be added to our slurry, such as fermented organic manure. FOM based microbial consortia is there, and we can directly spread broadcast these compost, or we can say that FOM, fermented organic manure, along with microbial inoculants. We can directly spread all these in our field, in the standing crop, and they have different microbes along with the carbon sources, and they can, when these microorganisms flourish in our field, they can degrade these residues, into simpler compounds, these lignocellulosic materials. And in this way, another method that is of liquid-based microbial product or consortium is there that can be sprayed into our field. We can spray and then incorporate the same residue in our soil that will help in the fast degradation or in maintenance of soil health, and even they can increase the soil organic matter, and you will find that yield will increase after some time if you repeat again and again 2 and 3 years. They will increase the soil nutrients, soil microbial biomass, and even help in the improvement of soil structure. So now question is that what we are doing in our university. You know, agriculture based mainly lignocellulosic biomass that can be degraded into simpler compounds. So biological biodegradation or degradation that is both economic and ecological reasons has become an increasingly popular alternative for the treatment of agriculture, industrial, organic as well as the toxic waste. You can see in this diagram, these are some fungi in our class. They are isolated from different locations, different autochthonous sites, different sites, and these are the hyper lignocellulosic producing enzyme fungi are there, and these fungi actually they secrete or produce lignocellulosic enzyme, cellulose, xylanase, lacase, lignin peroxidase, etc. that can be sprayed on the field, in the field where they proliferate, and again they help in the degradation of these lignocellulosic material. How they work, we have already discussed that these lignocellulosic biomass is there, and this can be degraded with the help of the lignin peroxidase lacase, particularly lignin or lignases. They can degrade cinnamyl, cinnapyl, etc., and then hemicellulose and cellulose slowly and slowly, and ultimately they lead to the formation of these monomeric units such as glucose, xylanose, arabinose, etc. So fungi, why we select fungi? Fungi actually they are the most active decomposer. They are unique in nature. They can survive in different ecosystem and can colonize over a wide range of agriculture residues. And these fungi have extraordinary catalytic capacity because they can colonize over a wide range of agriculture residue. And they secrete diverse extracellular enzyme. And these extracellular hydrolytic enzyme have breakdown capacity to break different polymeric substances into monomeric substances, either by hydrolysis or oxidation. Actually, these are again reabsorbed by these fungi. You can see here that these are some fungi. They are highly proliferative and they are fastidious in nature. They can adapt a wide range of agriculture residue, mean they can grow over a wide range of agriculture residue. And they can be specifically suited for the task of biodegradation because fungi have hyphal structure, and these hyphae mean just like they spread or over a wide range of surface, and they can grow over solid substances, and then they secrete enzyme, and these enzymes they are usually lignocellulosic in nature, and they can degrade a wide range of polymeric materials, into simpler one. Even then, they require very low amount of nutrients. Actually, we have to provide some conditions such as moisture. And even then, they can synthesize many commercially useful products from agriculture and other waste. You know, white rot fungi, that is very popular. Polyporus, lentice, coroides, Serenima maxima, etc. So they have a wide range of activities are there, and many biodegradation or lignocellulosic enzymes was observed in different studies in fungi, as we have already discussed in Aspergillus, that is found on onions, Trichoderma vidi, Trichoderma hergenium. They exhibit lignocellulosic activity. So cellulase system in fungi is considered to comprise three hydrolytic enzymes mainly. Cellulase means there are three main hydrolytic enzymes are there. Endo, 1,4-beta deglucanase, that can cleave beta linkase between two glucose units that constitute the cellulose. Then exo, beta 1,4 deglucanase, that can release cellulose from either of the non-reducing or reducing end of the cellulose, and beta glucosidase, which releases glucose from cellulose and short chain oligosaccharides. Here you can see cellulose degradation by cellulose enzyme complex. These are the leaf, plant cells, cellulose microfibrils. This one is microfibrils, and these three main enzymes, which are exo, endo, and cellulose, they particularly play an important role in the degradation. They can break these cellulose complex chains or long chain of polymer into simpler one, and ultimately they form monomeric unit of glucoses. Initially, if we subject microbial decomposition, when subjected to microbial decomposition, hemicellulose degrade initially at a faster rate and are first hydrolyzed to their components sugar and uronic acid. After that, they are converted into soluble monosaccharides in the form of either xylose, arabinose, galactose, and mannose, first degraded or hydrolyzed by means of extracellulase enzyme and then utilized by microbes. So different types of enzymes are involved in this process. They are endo and exo. Both type of enzymes are there, which can easily cleave these building blocks into simpler one. Here you can see hemicellulose degradation. Actually, what happened during this process? Xylanase, with the help of xylobiose, xylo-oligosaccharides, and exo and endo enzymes are there, which are responsible for the degradation of hemicelluloses into simpler unit. They can cleave the beta glucoside 1,4-beta linkase of complex polymeric chains into simpler one. Lignin that ranks second only to the cellulose as the most abundant naturally occurring polymer in the biosphere. Lignin actually is a complex, large molecular structure which contain cross-linked polymer of phenolic compounds, and these phenolic compounds are not easy to degrade into simpler one. They can take some times. Actually, these lignocellulosic materials, they are present in the primary cell wall, where they impart structural support, impermeability, and resistance against microbial attack. Basically, lignin contains three types of propionic alcohols, which we have already discussed in our previous slides. That is coniferyl alcohol, fumaryl, and synapyl alcohol. So fungi are also micro and microtype fungi are there, which are predominant, and they are effectively involved in degradation of lignin. Actually, microfungi, macrofungi are of two types according to their decay patterns, that is brown rot and white rot. The most effective lignin degrader in nature are white rot fungi. That is actually we can say that these are effective against lignin, that is white rot fungi, that have ability to degrade complex recalcitrant organic molecule into simpler aromatic residue or its constituents, either actually in aerobic conditions. Then microfungi, they degrade lignin under both aerobic and anaerobic, that is facultative anaerobic conditions in the environment, and ultimately they results into the formation of quality manure and humus. So fungi are the potential producer of lacase, manganese peroxidase, lignin peroxidase. These are the primitive enzymes, lacase, manganese peroxidase, and lignin peroxidase, which can modify lignin, or they are mainly responsible for the breakdown of lignin. A much smaller group of filamentous fungi have evolved with the ability to break down lignin, the most recalcitrant component of plant cell wall. We have discussed on white rot fungi, that is Phanerochite, Chrysosporium. They are actually most efficient in the degradation of lignin, and other fungi which are involved in lignin degradation. They are Phanerochites, Chrysosporium, and Pycnoporus cinnamarianis. These are some fungi which are used as a microbial inoculant for the in situ management. We can manage these residue both ex situ as well as in situ. Ex situ of the site, in situ on the site. And these fungi have ability to produce enzymes and metabolites, which can increase or fasten the process of decomposition. These inoculants, if we use these fungi as a monoculture, even though they produce these lignocellulitic enzyme, but when we mix them, if they are compatible with each other, then they have more pronounced effect, and they fastens the reactions, or we can say that degradation process. Actually, sometime what happen, these fungi and bacteria we can mix in a specific proportion to increase the degradation process, and if the process increases, then we can what we can conclude, we can conclude that if the process is fast, then they can accelerate the study composition and increase crop productivity. How they increase crop productivity? Either by adding some micro and micronutrients, which will improve the soil structure, soil nutrient capabilities. So if we have more than two fungi or microbes in a specific proportion, if they are compatible with each other, then we call them as a microbial consortia. And these microbial consortia can be applied as liquid or solid formulation. Microbial consortia can be applied in the field by mixing with the compost, or even as in the form of liquid or in solid state. If they are compatible with each other, that is another way. So many scientists conducted experiments to prepare consortia for fast degradation of rice and other residues. These are such as CDM, cellulose degrading microorganism. Even though Aspergillus, Bacillus species, they perform better during the degradation of rice straw. There are so many studies are there. So microbial in situ mean on site, and we spray these microbial inoculants either in the liquid form or in the solid form in the field to manage different crop residues. When they degrade these residue, it means they can increase the nitrogen, phosphorus, etc. in the soil and significantly increases their proportion in the soil and help in the maintenance, or we can say that they can perform for good soil health. There are so many studies are there. What we have done, we isolated many fungi and bacteria and then mix them whether they are compatible or not. After that, a lot of studies have been done either in laboratories, in pot houses, and ultimately in the field, mean from lab to land. What we have done here, you can see these are some microbes and these fungi, and these fungi when mixed in a particular proportion, after that they are added to these flask in laboratory conditions, and we have observed many datas or decomposition rate. Then ultimately in the land where we multiplied our consortia, and then it was spread in the field, and after that they are incorporated into the soil where we can enrich these particularly our consortia, which like these agriculture residue as a food, and then they degrade these agriculture residues. What we have observed after that, physical properties of soil, soil structure, soil moisture, soil temperature, and then chemical properties, whether what are the effect of these microbial consortia on the nutrient availability, soil organic carbon, and even microbial activity was also checked, such as soil cellulase, FDAs, FDA hydrolases, lactases, phenol oxidases, peroxidases. These are many different enzymes, dehydrogenases, etc. And we found that these activity was increased after some time, and ultimately we found that these microbes, they play a significant role, and even then they fastened the degradation process or decomposition process, and in this structure ultimately what we have done for 5 to 7 years, and ultimately found that after incorporation, lastly the recommendation of HAU biodecomposer, which is an intervention, only intervention for pedestal management in combined harvested field. Actually, we cannot say that these microbes only alone can degrade these residue, but this one is an intervention, and this intervention says that if we have 1 liter of HAU biodecomposer, we have to mix this in 5 liters of water in our fields, where we add 100 gram of urea, lime, jaggery, and besan, and then mix the and put the same for 72 hour under steady condition. After that, we mix the same into 200 liter of water, and ultimately we spray the same, whatever maybe we have instrument on this combined harvested rice straw, and after that we have to incorporate the same is residue into the soil, and then we can go for seedlings, and later on if we study the soil structure and soil microbial populations, chemical, physical parameters. So in this way, we have developed the HAU biodecomposer, which is an intervention for pedestal management. There are so many future perspectives are there, implementing more sustainable and comprehensive waste management practice, such as recycling, compost, safe disposal of the hazardous materials, and in this way we can help mitigate the environmental impact of agriculture waste and promote zero waste economy. encouraging collaboration between farmer, researcher, policymaker, and the public to bring awareness about the importance of proper agriculture waste management and potential benefit of sustainable waste to resources solution which can drive meaningful changes in the sector using genetically modified strain of microbes, some effective additives, and advanced technology can enhance the conversion of waste to resources. With the help of these, composting duration can be shortened. Increased collaboration between academia, industries, policymaker to address the barriers. The government should support composting initiative by offering financial assistance such as loan, grant to the farmer, setting up of composting facilities, sustainability, employment opportunities, and upliftment of rural communities can be done. This can encourage farmer to organic farming practices by emphasizing the use of compost as a natural soil conditioner and fertilizer. Bioreactor installation should be promoted as it can control the release of toxic gases, which can prevent these toxic gases to be released into the environment. And even though we can use charcoal in as a spotting, we can say that if we have charcoal, we can add or we can multiply these consortia, and along with charcoal, if it is sprayed into the soil, it can absorb carbon. So thanks to all for your kind presence. Hello.
Thank you so much, sir, for the insightful and thought-provoking session. We are extremely grateful for your valuable time and for sharing your vast knowledge and experience with us. We will now be moving on to the Q&A and interaction session. Before we begin, I would like to briefly share a few instructions for the participants. Those who are having questions or doubt are kindly requested to raise their hand. When you are invited to ask your question, please unmute yourself, turn on your camera, briefly introduce yourself, and then ask your question. We request you to keep your questions short, clear, and relevant to today's topic. Participants may also post their questions in their chat box. Ms. Simran will be handling the Q&A session and will call out the participants one by one. Over to you, Simran, please. Simran, are you there?
Two participants raised hand. Actually, Pooja Kumari and one more.
Yes, sir. Sir, we are having question from Bilal Ahmed. Bilal, please unmute yourself and ask your question. Bilal, unmute yourself. Okay, I think he may be. Bilal, unmute yourself and ask your question.
Good evening, sir.
Yes.
Myself Bilal Ahmed from Karnataka. Basically, I am from
बिलाल, आपकी आवाज नहीं आ रही. Repeat your question again. Bilal Ahmed.
Yes, sir.
Again, please repeat your question.
Yes, sir. I am audible.
Yes, now you are audible.
Yes, sir. I am from Karnataka.
Okay.
Suggest me a way to efficiently manage the sugarcane waste after its harvest.
Okay. From where we have to wait for the sugarcane residue management. Again, you can ask.
Hello, Dr. Jagdish.
सर आवाज नहीं आ रही आपकी। हेलो। चल चल अंदर चलो।
हाँ जी डॉक्टर साहब, ऑडिबल डॉक्टर साहब प्लीज।
हेलो। हेलो।
हाँ जी डॉक्टर साहब,
सर आवाज
अब
नहीं आ रही
आपकी
आपकी।
आवाज आ रही है।
आवाज आ रही है ना? हेलो।
हाँ जी सर, अब आपकी आवाज आ रही है।
ओके।
यस सर।
हेलो।
हाँ जी सर।
हाँ अब बोलिए बेटा, क्वेश्चन क्या है, आपकी क्वेरीज क्या है?
Sir, I am co-host. I am handling the query session. Piyush Kumar Yadav, you can unmute yourself and ask your question.
पीयूष।
Okay,
पीयूष इज देयर?
Piyush is there. Yes, please unmute and ask your query. Piyush Kumar Yadav.
गुड इवनिंग सर। सर मैं गोरखपुर का रहने वाला हूँ सर। सर मेरा ये क्वेश्चन है कि हमारे पास मतलब लैब में बहुत ऐसे प्रयोजन हैं, जैसे कि किसी माइक्रो ऑर्गेनिज्म को हम ग्रो कराते हैं। मतलब ऐसा कोई उपाय है जो किसान आराम से कर सके, क्योंकि किसान लैब टू लैब में थोड़ा कतराता है कि वो मतलब किसी लैब से ना हो करके ही वो खेत में ही डिकम्पोज हो जाए। ऐसा कोई तरीका?
लैब टू लैंड की हम बात कर रहे हैं। अभी हमने जो बात की ना, कि अगर हम अपनी पॉपुलेशन इंक्रीज कर देंगे, अगर नेचुरल फार्मिंग की बात करते हैं हम, तो नेचुरल फार्मिंग में क्या है कि माइक्रोब दो तरह के हैं हमारे पास। हार्मफुल भी हैं हमारे पास
यस सर।
और बेनिफिशियल भी है। लेकिन हार्मफुल कितने हैं? 0.01% इज देयर,
बिल्कुल
एंड
बिल्कुल
मोर
सर।
देन 99% microbes are our friends.
यूजफुल।
And we have to enrich the population of these microbes in a particular field. And we can do this with the help of these microbial consortia. If we add microbial consortia first year, then second year, and then so on, after 2 or 3 years, what happen? They adapt that particular place. Mean they explore, just like succession is there from pioneer to complex climates. In the
यस सर।
similar way, we can increase or enrich this population. So farmer have to add these microbes in the field up to 4 to 3 years, 3 to 4 years, and only
सर
that
ऐसा कोई
we
प्रोविजन
can increase.
जिसमें टाइम मिनिमम हो, मतलब कम समय लगे?
कम समय लगे। कम समय का देखो, ये पार्टिकुलर लैंड टू लैंड डिपेंड करता है कि कहीं बार हमने जो काम किया है, एक दो विलेज है, जैसे गांव है हमारे यहाँ
जी
हरियाणा
सर।
में, हिसार के पास एक फार्मर है हमारा, काफी अच्छा 70 एकड़ में वो काम करता है, और अपने खेत में कोई पेस्टिसाइड और इंसेक्टिसाइड नहीं डालता, टोटली ऑर्गेनिक काम करता है वो। तो क्या
ओके
होता
सर।
है कि कई बार हम माइक्रोबियल पॉपुलेशन, जब हमारी जो एग्रीकल्चरल प्रैक्टिसेस जो होती हैं उनसे वो डिस्टर्ब हो जाती है पॉपुलेशन,
बिल्कुल सर।
तो वो फार्मर क्या करता है कि सारा का सारा ऑर्गेनिक बेस फार्मिंग करता है, नेचुरल फार्मिंग करता है। तो उसने लगातार 3 साल तक डिकम्पोजर को यूज किया यहाँ। तो उसके खेत में जब चौथी साल पांचवी साल हमने रिजल्ट्स देखे कि वहां पे जो पराली को जो है ना, वो क्या करते हैं, इनकॉर्पोरेट करते हैं। इनकॉर्पोरेट कैसे करते हैं? जैसे हमारा जो माइक्रोबियल कंसोर्शिया है हमने दिया, उसको उसने मल्टीप्लाई किया, मल्टीप्लाई करने के बाद में 200 लीटर पानी में मिला करके उसने स्प्रे कर दिया। और स्प्रे करने के बाद में मॉइस्चर जो है ना, अगर 60% है आपकी सोइल में, तो उसको इनकॉर्पोरेट कर देते हैं सोइल में। जब आप उसको इनकॉर्पोरेट करोगे, उसके बाद वो आपकी जो रेट जो होती है माइक्रोबियल जो ग्रोथ है,
रेट ऑफ माइक्रोबियल
वो
एक्टिविटी।
एक्सपोनेंशियल हो जाती है, और वो एंजाइम और मेटाबोलाइट ज्यादा सीक्रेट करते हैं। और जब एंजाइम और मेटाबो और लगातार 1 साल 2 साल 3 साल उसने प्रयोग किया, तो लगातार प्रयोग करने से क्या है कि वहां जो उनकी पॉपुलेशन है, जैसे हमारा सक्सेशन जो होता है ना, उसमें एक पहले एक कम्युनिटी आएगी, फिर सेकंड, फिर थर्ड थर्ड, तो वो वहां पर अडेप्ट हो जाती है। तो लगातार अगर आप ऐसा करता रहेंगे, और केमिकल का उसने यूज नहीं किया, तो उस खेत में मैंने देखा है कि उसको तीसरी चौथी साल जो है वो अपनी प्रणाली को जो है उस खेत के अंदर इनकॉर्पोरेट कर देता है विदाउट, तो भी वो जल्दी इजिली जो है वो डिग्रेड हो जाती है। बट
ओके
डिग्रेड कितने दिन में होती है? 30-40 दिन में होती है। लेकिन एक्चुअल में मिनरलाइजेशन जो मैंने कहा कि लिग्निन कंपोनेंट है, दैट इज द बेसिक
जी,
प्रॉब्लम, कि लिग्निन जल्दी से जो है ना वो टूटता नहीं,
डिग्रेड नहीं
डिग्रेड
होता।
नहीं होता। तो उसको क्या है कि उसको डिग्रेड होने में टाइम लगता है। इवन देन मैंने कहा था कि साल भी लग जाता है, अगर आप देखें तो। वो
यस,
डिपेंड
इट्स
करता
वेरी
है कि वहां पर जो
कॉम्प्लिकेटेड।
अवेलेबल जो सॉइल है उसमें माइक्रोब्स कितने हैं, वहां उनकी पॉपुलेशन कितनी हुई, वो सारी कंडीशन जो है ना वो भी जरूरी होती है माइक्रोब्स के लिए। तो लैब के अंदर हम कंडीशन जो होती है स्पेसिफिक होती है, लेकिन फील्ड के अंदर इट इज वेरी डिफिकल्ट टू मेंटेन these conditions in the field,
यस, ऑफ कोर्स, बिल्कुल
because
सर।
there is a so many adults are there, climate condition, water conditions, even chemical herbicides, and there are so many factors are there. That's why we are facing these problems.
थैंक यू सर।
Next one.
Sir, we are having question from Arun Dayal. Please unmute yourself and ask your question.
Next one.
Sir, we are having question from Arun Dayal. Arun Dayal, please unmute yourself and ask your question.
जी जी,
डॉक्टर
शुक्रिया।
अरुण दयाल, यस प्लीज।
डॉक्टर साहब नमस्ते। मेरे पास दो सवाल हैं। एक पहला सवाल उस लेक्चर से रिलेटेड है जहाँ पे हमने नेचुरल फार्मिंग के बारे में बात की थी, और वहाँ एक सजेशन था कि पराली जैसा प्रोडक्ट जलाने की बजाय उसको हम मल्चिंग के लिए यूज़ करते हैं। तो वो मल्च कितना परसेंट ऐसा है जो रीयूज हो जाता है मल्च में, और बाकी ज़रूरत पड़ेगी कि हमें उसको प्रोसेस या डिकम्पोज करना पड़े? ये पहला सवाल है। दूसरा सवाल ये है कि जो एचएयू ने बायो डिकम्पोज़र बनाया, या फिर ऐसे और जो डिकम्पोजर्स अवेलेबल हैं, हम अभी भी देखते हैं कि किसान तो जलाने, वो इजी है क्योंकि जला दो, क्योंकि वो प्रैक्टिस भी है, नॉर्म भी है, चल रहा है। आपके हिसाब से जो अडॉप्शन रेट है या फिर अडॉप्शन का जो प्रैक्टिस है, उसमें चैलेंजेस क्या हैं? मतलब ऐसी क्या दिक्कत आ रही है जिसके अंदर ऐसे डिकम्पोजर्स का यूज़ अभी भी एक्सपेक्टेशन से कम है?
सर, पहली बात मैंने आपको पहली स्लाइड में ये बताया था कि इग्नोरेंस, अवेयरनेस, नॉलेज और सरकार का सहयोग, और हमारे पार्टिकुलरली जो फार्मर हैं उनका सहयोग चाहिए। दूसरी बात, हरियाणा के अंदर अगर आप रेट देखेंगे इस साल जो है बहुत अच्छी रेट रही कि लोगों ने पराली बहुत कम जलाई है। इसके लिए हमें फार्मर ट्रेनिंग है, और आपके जैसे ये ग्रुप है ना, जैसे सर ने बनाया हुआ है अजय ने, वो ऐसे ग्रुप नॉलेज अवेयरनेस अजय सर जो कर रहे हैं, इस तरह के जो कार्य हैं ना वो किए जाए, लोगों को ज्यादा से ज्यादा अवेयर किया जाए, तभी इस प्रॉब्लम का हम सॉल्यूशन कर सकते हैं। दूसरी बात, डिकम्पोजर जो एचएयू की बात थी, एचएयू का जो डिकम्पोजर है दैट इज एन इंटरवेंशन, कि आपके पास कई कंपोनेंट है, आपके पास मशीनरीज भी हैं, हैप्पी सीडर है, सुपर सीडर है, या दूसरी मशीनरीज हैं, उसके साथ हमने एक एज एन इंटरवेंशन है, वो एक इंटरवेंशन इन द सेंस कि वो उस चीज को फास्ट कर देता है। और क्या था सर आपका क्वेश्चन? हेलो हेलो हेलो।
हाँ जी डॉक्टर साहब,
हाँ
आई
जी सर।
होप क्यूरी ऑफ डॉक्टर अरुण
सर वो
दयाल हैज
पहला
बीन रिजॉल्व्ड।
सर
यू
पहला
कैन,
पार्ट वो था
यस
जिसके
यस,
अंदर
नेचुरल फार्मिंग की आप बात कर रहे थे।
सॉरी डॉक्टर साहब
हाँ हाँ, नेचुरल फार्मिंग
कि
मल्चिंग की बात हुई थी।
हाँ जी, मल्च के अंदर रीयूज करना वर्सेस डिकम्पोज करना कितना परसेंटेज है?
सर इसके लिए आजकल वो रीजनरेशन एग्रीकल्चर कर रहे हैं ना, कुछ रिपोर्टें आई है, कुछ लोगों ने काम किया, हमने अभी तक ये इतना काम किया नहीं। तो नेचुरल फार्मिंग के लोग हैं ना, वो ज्यादा बता पाएंगे। आजकल एक रीजनरेटिव जिसको हम कहते हैं ना, सोइल को बिल्कुल वो जीरो टिलेज की तरह रखते हैं, काफी लोग काम कर रहे हैं। स्टैटिस्टिक्स डाटा नहीं, अभी कल ही एक शिक्षा से आती है काफी अच्छा काम है। तो 10 लाख जीरो टिलेज की तरह का काम है। तो मनीष है, और तो अटल अवर फार्मर लैक्स जो है ना, उसमें अगर एक आपको मैं कि जो है ना कवर फार्मर जो कवरिंग करते हैं उसकी बात कर रहा हूँ, वो मल्टी इश्यूज कवर हाउ दे वर एबल टू रियलाइज इकोनॉमी, इसके ऊपर काम कर रहे हैं लोग। तो इसका हमारे पास कोई इतना ज्यादा डाटा नहीं है। असल में ये हमारे फील्ड थोड़ा सा ऑर्गेनिक जो नेचुरल फार्मिंग पर काम करते हैं वो लोग ज्यादा। आवाज आ रही है क्या सर आपको? हेलो,
हाँ जी
आवाज
सर,
नहीं?
अभी आ रही है।
अभी आवाज आ रही है मैडम?
हाँ जी आ रही है सर।
मेरे प्रॉब्लम तो नहीं है नेट की तो कहीं भी।
तो थोड़ी आपकी आवाज ब्रेक डाउन हो रहा है, उसका ब्रेक डाउन हो रही है सर।
नहीं, ऐसा तो नहीं है मैडम। रेंज तो काफी है मैडम यहाँ।
बीच में सर आवाज थोड़ी लो हो जाती है।
एक्चुअली में मैडम आज एकदम मेरे को जो है ना, मेरे पास वो नहीं है, ईयरबड्स वगैरह कुछ नहीं है, डायरेक्टली इसको मैंने लैपटॉप पे किया है। तो हो सकता है उसमें प्रॉब्लम हो, अदरवाइज तो प्रॉब्लम नहीं होनी चाहिए।
Okay, now you are audible, Prashad, and we can continue the Q&A session. And to add some points to the Dr. Arun Dayal query, like you have asked, like how we can manage the crop residue in the mulching, and specifically you have asked about the natural farming. So in that case, I think you are aware about and you have attended our last 3-day session, which are on natural farming. And the mulching is the major thing we are adopting in the case of natural farming, because whatever the residue is there, that is incorporated and that is biodegradable. In the meantime, when we are not adding the fertilizers into our field, the crop residue itself is the enriched with the different nutrients. So that will be added, and definitely our crop will get some nutrients, while growth will that will sustain our growth. Okay, that is the one point. Another one is like if you do the mulching, we have talked about the WAPSA in natural farming. The so mulch will be managed, that automatically help our growth, the growth of our crop. And the third point we can say, the mulching is also required to manage the sunlight. Like whatever the sunlight will come to our plants, that will also be trapped and that will be provided sufficiently to our crop growth. So these are the 2-3 points we can take out of this. And definitely the last point we can say, it is a alarming for all of us that burning of the residue is on peak, especially if you talk about the North India in the Delhi or in Haryana or the Punjab. So it is a very big question mark on the agriculture department as well as also the farmers, because it is contributing to the uh, environment pollution. So in that region, uh, given the example, last year we have so many cases of crop residue, but this year we have only 2 to 3 cases reported so far. So this is all about the awareness or the training of the farmers and the usage of crop residue. Like if that will be incorporated into the field, the crops will grow better. So likewise, we can understand the usage of crop residue, and definitely it will be helpful for the farmers to sustain the crops, and it will also be helpful if we adopt the zero tillage and other practices. So now we can move to the further queries.
Sir, nowadays there is another concept that is cover farming, which is an important and sustainable approach toward improving soil health, conserving moisture, and enhancing farm productivity. But in the present scenario, what happen? Scenario of climate variability are there, declining soil fertility, rising input cost. Cover farming, that is one of the most emerged as a practical and farmer-friendly solution. It play crucial role in protecting soil from erosion, improving organic matter, enhancing microbial activity, and suppressing weed particularly. Most importantly, it supports sustainable agriculture by reducing dependency on chemicals, as well as what we have gained knowledge from other audience. Only that is in our mind. So I think cover farming is one of most emerging field nowadays, and a lot of work need to be done in this field.
जी डॉक्टर साहब, definitely it is going to be a sustainable farming practice in the coming future, and farmers and the students whoever are here, they will adopt the core farming as well as the natural farming, so that we can be eco-friendly and can grow the better crops for the better health of the people. So thank you so much, Daksa, for addressing this query. And now we can move for other question. Vivek Yadav, please unmute and ask your query.
Vivek is there. Vivek, ask
I
your
think
question. What
he
is
is
your
not
question?
available. I request to all the participants, whoever raise their hand, it means they are desire to ask the queries. So please be attentive whenever, because we will call your name so that you can ask your query. So moving ahead, BU809473 student ID, please ask your query.
सर मेरे ख्याल में सबके क्वेश्चन जो है ना पूरे हैं, सबको आते हैं सारे। मोस्टली लोगों को पता है कि बहुत सारे डिकम्पोजर आ चुके हैं मार्केट के अंदर, और हमने बताया कि हमने इंटरवेंशन दिया है ओवरऑल, और हमने जो किया है वो हमने यहाँ पर आपके पटल पे जो है ना, मेरे को ये पता नहीं था कि आपको इतना बहुत अच्छा प्रोग्राम होता है, इतने पार्टिसिपेंट्स होते हैं। मैं तो जस्ट लिया, आपने कहा मेरे को सुबह, और मोमेंट्री अगर हम बात करें तो मैंने तो स्लाइड भी नहीं तैयार की थी सर। एक्चुअली जो भी जहाँ से आई आधा घंटे में तैयार की और आपको लेक्चर डिलीवर कर दिया। तो
नहीं, ये डॉक्टर साहब स्लाइड्स के ऊपर नहीं, ये तो आपका अनुभव, आपका 20 से 25 साल का जो क्रॉप रेजिड्यू पे है, तो वो बोलता
सर
है। स्लाइड्स तो क्या है, बच्चों की हेल्प के लिए है कि भाई आपने लिखा है, थोड़ा अटेंटिव रहते हैं बच्चे। बाकी जो आप बोल रहे हैं, जो अपन
नहीं
को
सर,
मैसेज
मेरा
कन्वे कर
एक्सपीरियंस
रहे
तो
हैं।
इसमें 7-8 साल का है,
हाँ जी, हाँ जी।
रेजिड्यू का तो 6-7 साल का ही है। हाँ
जी डॉक्टर साहब, फिर भी जो है बच्चे इंटरेस्ट ले रहे हैं और डेफिनेटली जो क्वेश्चंस पूछ रहे हैं। अभी एक जो है क्वेश्चन आया है मेरे पास में डॉक्टर साहब, विष्णु प्रताप जी हैं, and he is from Kanpur, Uttar Pradesh, VNMKV University, Maharashtra. So his question is, the amount of lignin in wheat straw is 15%, and in rice straw it is also 15%. So it is very hard to decompose the lignin. So I cultivate both crop one by one in continuous season, once like wheat and then rice. So after I sow maize that contain only 7% lignin. So what process I should follow to control the lignin? And please tell me what component I can apply to control it. It is very interesting question, in-depth question asked by Vishnu Pratap.
Actually,
Dr.
this question
Yes.
is very interesting, and presently we have started a new experiment in our research that is the rice wheat cropping system is there, in which there are two crop, basically wheat and lignin, wheat and rice, which contain lignin up to 15 to 20%, and wheat straw actually have lignin about 16 to 80% is there. So lignin in the wheat straw is moderate. That mean moderate mean making it comparatively easier to decompose than the rice. because rice contain, if we go for private basis, lignin content is actually 18 to 25% is there, and typically it contain 20 to 22% lignin in rice straw as compared to wheat straw, that is 16 to 80%. That's why we can say that it is very easier to decompose in wheat than the rice. Rice straw has a higher lignin and silica that make it more recalcitrant and slower to decompose. That is the main reason. So this is the main reason, sir. And they require lignocellulitic fungi. And presently we are doing on this alternative system, that is rice wheat cropping system, and the same microbial consortia, either from our university, from PUSA, and other institutes, we have applied in this year. So we can say that we are doing or contributing to increase the research on these recalcitrant and slower biodegradable compounds by using these consortia. That is the main reason, sir.
Okay, thank you, Daksha. So we have one more query from Venkatesh D. Babu. You please unmute and ask your query.
Sir, good evening, sir.
Good evening.
Sir, you have given an excellent presentation, sir.
Thank you, sir. Thank you.
Sir, I am working as assistant professor in Department of Crop Physiology, sir.
Okay.
Anguru. Uh, Sir, my question, you already answered that question. Is there an existing consortia that can decompose the paddy straw actually? That is the major issue now.
Okay.
Is there up to date any consortia there that can able to decompose in a faster way? Less
Okay,
duration.
But we can say that, sir, this one is, I think this one is a component. Intervention is there.
Okay,
This
sir.
one is one of components, not
Sir.
completely it can degrade. If we use it with other mechanization such as happy seeder.
Happy seeder, yes, sir. Yes, sir. Happy seeder, the baler is
Then,
there. Baler, so many are there. Because of reducing in the reduced polled, what you can say, animal strength from the farmer side. So there is lot of straw is going to be waste in the fields, sir. Actually, they are going for burning. So there is no alternate for them. Actually, they are not maintaining any livestock. If there is any livestock, they would have used that straw actually.
Rightly said, sir. Actually, we are doing nowadays work on fermented organic manure along with this microbial consortia. That
Yes,
can
sir,
help,
yes.
or we can say that it can increase these rate of decomposition of these
Yes, yes.
recalcitrant compounds.
Yes, sir. Yes, sir. That is the main issue, sir. Actually, in future burning issue also the same because the farmers are not in a patience to maintain the livestock. Actually, if they maintain livestock, it will be to some extent it will be useful for the livestock. Actually, but
That's
they are
why
not
people are doing natural farming, and our government is also increasing this concept.
Yes, sir. Yes, sir. It is a huge climatic problem also. It is creating so much of climate change in Delhi because of the Punjab and the UP. They are burning the crop residue. So because of there is a law enforcement also, still there is burning is going on actually. That consortia, if you discover that consortia, that will be a great discovery, sir, for Indian agriculture, crop residue management. So other side we have the municipal solid waste also. Again our agriculture waste is another problem. It is a rising problem again.
Sir, that's why I am saying that nowadays, if we have a household product or agriculture household product and other waste are there.
Yes, sir.
So first of all, what we can do, we can ferment them.
Yes, sir.
And that one is the fermented organic manure. That
Okay,
can
sir.
be added to these microbial associates, which is very, we can say that helpful in increasing the soil health by
Yes, sir.
increasing particularly carbon.
Yes, sir. We have to explore many ways to what extent we can reduce the straw, sir. Many ways we can convert waste to energy, like waste to energy. Some straw we can use in other products,
biofuel
making
and
other products,
etc.
etc. Making other products which are which will be more useful. Some other like making ropes or something, some other products. Thank you, sir. You have given excellent
Okay.
presentation, sir. Our coordinates are also given a glimpse of that natural farming with residue, crop residue management very excellently. He has given 2 minutes glimpse, sir. Very, very excellent, sir. That is the burning issue now, and people has to move into the natural farming to some extent so that all these problems will reduce to some extent.
Thank you so much, Dr. Venkatesh. So now we can move to the next question. The question is from Siri Karini, and she is asking. Her question is, she is the UG student from Telangana. All these residue management techniques or the decomposition techniques work in all the conditions, or the we can say agroclimatic conditions, irrespective of the natural condition of the soil, like in that of arid and semi-arid regions. So, her question is like these are region specific, or whatever you have taught during the session, or these are applicable in all the agroclimatic zones, like in Telangana, in Haryana, in Punjab, wherever we are talking about the agriculture. So please focus in that, sir. Dr. Jagdish, am I audible to you? Hello.
हेलो
हाँ जी डॉक्टर साहब, अभी आ रही है
हाँ जी
आवाज,
सर,
प्लीज।
2 मिनट में बाहर चला गया था सर कहीं। हाँ जी सर।
हाँ जी डॉक्टर साहब, Siri Karini is asking a question, and she is UG student from Telangana. Like whatever the decomposition techniques you have told into the session, are these applicable to all the agroclimatic zones, irrespective of the soil natural conditions, or these are region specific?
No sir, these are applicable to. Actually, we have done these experiment only in Haryana and Punjab. So in our agroclimatic condition, they are doing well. But I can't say what is the agroclimatic condition in Andhra and other states.
हेलो।
हेलो।
हाँ जी डॉक्टर साहब।
सर जो हमने काम किया है वो यूजुअली बेसिकली हमने तो हरियाणा में किया है, बट आई थिंक इट कैन बी एप्लीकेबल टू ऑल एग्रोक्लाइमेटिक कंडीशन.
Okay.
As
I
we have
also.
done these work from lab to land and even in hothouse conditions and pit out conditions.
And Dr. Saab, and for the information of the students, like IRI PUSA have like produced the biodecomposers, and our own university CCSHU Haryana produced biodecomposers. Likewise, all the state agriculture industries and the central agriculture industries are working on this. Okay, because crop residue burning is a very hot topic, and it is very problematic for our environment. So definitely these are applicable to all the agroclimatic zones, and there are different practices, but the major practices like we have talked about the in situ and ex situ management of the crop residue, so that are applicable to everywhere. The methods may change, but the core principle, or we can say the management we
The
have to
core
do is.
principle is I think the same. Recently, you know that IARI has developed microbial capsule based consortium for decomposition of pedestra, which is working actually for short duration time. That is named PUSA decomposer. Similarly, another ICR institutes, ICR and BAIM, MAU, They also developed under the name of biocompost, which is mainly consists of Trichoderma viridi, Aspergillus niger, Plutorius florida, and Phanerochaetes chrysogenum. Another on the same line, ICAR Tamil Nadu, that is also developed a TNAU biofertilizer and biodecomposer consortia, which is mainly consists of bacteria, fungi, and actinomycetes. Another that is ICAR Institute IISS Bhopal, that also recommended phospho sulfo nitro compost. And another institute that is ICAR AAU and Johrat, they also suggested bio-enriched compost, which is in this the final compost is amended with the rock phosphate, 1%, and microorganism like Pseudomonas, Azospirillum, Azotobacter. So ICAR NRRI Katak, that also tried with 3 consortia for rapid ex situ as well as in situ decomposition. So these biodecomposer are available in different agroclimatic zone, and they are working on the same principle. So we can say that each and every consortia work in a similar way in different agroclimatic conditions.
Thank you, Dr. Saab. So we have one more question on the same Sri Karini, and she is asking, like, we follow the integrated pest and disease management practices, and in that case, we remove disease and pest management. So in that practice, we remove the infected plants or the plant parts. So should we destroy these plant parts, or if we should incorporate into the field, or we have to remove from the field? So before Dr. Saab, want to answer that query, we definitely need to remove the infected plant parts away from the field. Otherwise, there will be development of some microbial family colonies, and that will be problematic for our crop. Whenever you find some infected or the diseased plant parts, you please remove those away from your crop. And if there are any additional point to it, please you can guide us, Dr. Saab.
Sir, actually, you say that we need to remove these pathogenic plants. That is the best process. Otherwise, what happen in during the composting process, we are going for composting. That is particularly acceptable. In that condition, we have discussed on the composting phase, that is the thermophilic phase. These harmful microbes cannot survive, and they are. I can say that they can be killed from different metabolites which are secreted by these microbial concepta, and it depends on the nature of have either bacteria, parasites, fungi, etc. That depend on that condition. So that one is the good question from INM, how to remove these pathogenic plant, and we have to do some work on this. I think we have to work.
Okay. And thank you, Dr. Saab. So we have like 4 to 5 more questions that are very relevant to the topic. So if you please allow us, can we carry forward this Q&A session?
You can carry, sir.
Okay, okay. Thank you so much, Dr. So there is one more question from Yenesati Shivani, and she is a MSc graduate, MSc scholar. And her question is, can crop residue management help in reducing fertilizers use cost for farmers? How it can be helpful for specifically for the farmers?
Actually, they are helpful to us because these microbes have some plant growth promoting activities are there, and we are doing in this that I have already told that Trichoderma hergenium and Trichoderma viridi, they have more than 95 peptides which are beneficial, and even these metabolite help in the seedling or in germination, or even they are very helpful. They can control, or they can be used as a biocontrol agents. And we work on this. Actually, we are doing on this parameter. As our consortia have many of the PGPR activities like bacteria and siderophore productions, etc., which can remove the hazardous material as well as pathogenic microbes near to the rhizospheric zone.
Okay, thank you, Dr. Saab. There is one more question, and it is very important for the students. Like many students are asking the same question. It's D. Smith from Dhole, Pune, and his question is, Sir, I wanted to start an agricultural waste management enterprise. Like he's looking for entrepreneurship in this particular field. So, can you please guide to scale it, and which the specific area should he focus on? And in one additional question, can we earn carbon credit on agricultural waste management?
सर क्वेश्चन अच्छे हैं. अभी आपने विजिट किया है हिसार में ना, तो हिसार में आपने देखा होगा वन ऑफ आवर फार्मर और incubator is there in Chaudharywas village. What he had done, he collected different agriculture residue as well as cattle dung, and then fermented the same, and he make a product such as FOM, fermented organic manure, and he is selling the same, which also increases the carbon level of soil. As we have already discussed, that if we incorporate these microbes or consortia along with biofertilizer in fermented product or fermented organic manure, then we can increase, I think, carbon in the soil. That is very helpful. And he can become an incubator, and he can do his work or at any place that is accessible to him or us.
Thank you, Dr. Saab. And students, definitely this biodecomposer concept is new and very effective nowadays. So you can work on it. You can involve in some projects undergoing under your university as PI, and these are there. You can contact them and be a part of these projects. And you definitely, as an enterprise, you can start this on scaling this particular composers business.
Actually, what happens, sir,
There are a lot of things.
Sir, crop residue management and
yes, Dr.
You can say that if we have undecomposed residue that may harbor pathogens, that is the INM based practice, microbial decomposer, usually residue composting, residue in situ residue degradation. They destroy pathogen survival structure, and even they enhance the microbial dominance of these particularly added microbes or enriched microbes. They are so they can improve the soil structure, their physical properties such as soil structure, aeration, drainage system, and poor aerations and water losing favor the pathogen. You know that poor aeration and water losing actually they favor pathogens. So INM that reduce favorable condition for pathogens, but these decomposer, I think, help in we can say that maintenance of good soil structure and soil properties, soil moisture, aeration, etc. You know that plant develops systemic resistance, making them prone to infection, even pathogens are present. So I think these pathogenic plants we can either remove from the field, or if their litter portion remains, then there is no problem. Because integrated nutrient management actually They suppress plant pathogen indirectly by enhancing soil microbial biodiversity. So improve nutrient balance, promoting antagonistic mechanism, microorganism, and even then plant have their strengthening mechanism, defense mechanism system is there.
Thank you, Dr. Saab. So there is one more question from Sri Chandra, a third year UG student of agriculture. And his question is there any specific difference between crop residue management practices in the horticultural and the agricultural crops? Since their growing time and their different agricultural practices and the agroclimatic zones, everything is different in the case of horticulture and agriculture crops. So is there any major difference in the crop residue management also? Please guide, Dr.
Sir, actually, in horticultural area, I think we have not done so much far in these horticultural crop in decomposition process. Only we have done work on rice wheat cropping system only. That's why we have no idea about the horticulture, particularly in situ decomposition.
Okay, dear participant, definitely we will answer this query and you can ask the same query into the feedback form also. So no issues, doctor. Now we are moving to the end of this session and we have the last question from the student with ID number 9473 from BUT Banda. Please, who has raised the hand, please unmute and ask your query. Okay, let me tell you all the participants, it is for your kind information. Your university coordinators are also here. Okay, so you please don't think that you can do such type of nonsense like you will raise hand and you are going to disturb the audience. Okay, please be attentive and attend all the sessions. These are very informative and definitely these are going to shape and reform your mind makeup and whatever you are doing in your respective degrees, you will learn something apart from that. Okay, uh, because you know this is very burning topic and we are here on the same platform talking about the crop residue management, and I can see many students are just here and they are not very attentive. But let me tell you, the questions will also come from this class, and you have the examination at the end of this training program. So please, it is for your reminder. If you will be attentive, definitely it's not about exam, it's not about certificates. It's about your personality development and your knowledge enhancement. You will be in a better phase after completion of this training program. Okay, and we are consistently trying to aware you people and connect with the most effective and the experienced faculties from all the agricultural allied sciences. Like today we have Dr. Jagdish Prashad with us, a microbiology expert, and he has the expertise in crop residue management, and he has also been the active member, or we can say the torch bearer in the biodecomposers development at the CCHHI. And yesterday we had Dr. R.C. Agarwal, DDG, Agriculture Education. Day before that day, we had Dr. O.P. Chaudhary, beekeeping experts. So we are going to have more experts in the coming days. Like more Dr. A.K. Nayak, DDG, NRM, ICR is going to be there very soon. And many more eminent speakers will be there. Some eminent speakers from the international scientific community also. So please, you all are requested to join the classes because there are 700 participants registered for this training program. But as I can see today, there are only 400 or the 450 sometimes joined. So it will be problematic if you will not join because this attendance is being submitted to the university coordinators like Honorable Registrar and the Honorable ICR nodal officers. So please be cooperative and learn something from this platform. It is for you people. Okay, and it is very important to ask the queries also. because the expert also came to know that how much effective the class was and how he was able to create some impact in your learning and your knowledge is enhanced or not. So thank you, Dr. Jagdish Prasad. With this note, and Simran and Dr. Pooja, please carry forward and conclude the session. Thank you so much.
Thank you, sir. On behalf of the Agri SAARTHI team, I would like to express our heartfelt gratitude to Dr. Jagdish Prasad, sir, for delivering such an enlightening and impactful session on crop residue management. Once again, thank you, sir. I also thank our organizing director, Mr. Ajay Kasana, sir, Chief Secretary of Agri SAARTHI, for his continuous guidance throughout the session, which helped in the smooth conduct of today's program. I thank all the participants for your enthusiastic presence, active engagement, and meaningful questions, which made today's session truly interactive and successful. Finally, my gratitude to the entire Agri SAARTHI team for their continuous effort in organizing this 30 days National Agricultural Training Program and for providing such a valuable learning platform. Before we conclude, I kindly remind all the participants that the attendance form has been shared in the chat box and in the group. Filling this form is mandatory and it will be active for only 30 minutes. Thank you all once again for joining us today. We look forward to your participation in the upcoming session of Agri SAARTHI KRANTI 2026. Have a wonderful day ahead. Jai Hind.
Thanks, sir.
Thank you
Dr.
so
Ajay,
much, Dr. Saab,
thanks
and thank you, Dr. Pooja.
for your sincere support. Actually, if you have such a lecture, then you have to give us some time for the preparation. आज का लेक्चर जो रहा एक्चुअल में अगर आप मेरे को टाइम देते ना बड़ा लंबा
Definitely, Dr. Saab, we can understand that
आज
you
तो
have
आपने
the busy
बस
schedule. So
मैंने कह दिया और आज सर आज एक्चुअल में हम सारा दिन बिजी थे सर सारा दिन जब आपके फोन आए
हाँ जी,
तो
हाँ
मैं
जी,
रिप्लाई भी नहीं कर पाया आपको कई समस्याएं थी बट कैसा रहा सर आपका
definitely, Dr. Saab, it was a great session because we are getting a lot of appraisals from the students. ओके एंड बहुत ही अच्छा जो आज का लेक्चर रहा और बच्चों ने जो है क्वेश्चंस पूछे उससे आप समझ सकते हैं कि इफेक्टिव था सेशन और बहुत ही अच्छे से जो है
पार्टिसिपेंट्स काफी हैं सर आपके यहाँ
हाँ जी डॉक्टर साहब और काफी अच्छे बच्चे हमारे पास में जुड़े हैं डॉक्टर साहब बीएटी बांदा से बीसीएस यूएस पौड़ी
एक्चुअल
गढ़वाल
में
से
सर चलो मैं
यूएस रायचूर
फोन
से
पे बात करता हूँ सर आपसे
हाँ जी हाँ जी ओके
ठीक है
थैंक यू
सर
सो मच डॉक्टर साहब थैंक यू सो मच फॉर गिविंग सच ए informative lecture. It was really in-depth session.
थैंक यू सर
And
थैंक यू
I request all the participants, the attendance come feedback link has been posted into the chat section. So please, all of you are requested to copy and paste it into your Chrome or your browsers, and you can leave the meeting. It is ended. So thank you so much for participating and be there for the coming sessions with the same enthusiasm and energy. Definitely, you will learn something. Thank you so much. And now the meeting is closed. Jai Hind.